All right, so welcome everyone to today's seminar. It's my pleasure to introduce our speaker for today. Dr. Jamie Hall is a Tenure Track Fellow at the Institute of Infection, Veterinary, and Ecological Sciences at the University of Liverpool, where he studies the evolution of bacteria, parasites, and viruses. He is particularly interested in using experimental evolution to study how microbes evolve in complex environments. And I do want to give a huge thanks to Dr. Hall for agreeing to this virtual visit. He is in a very different time zone, and currently for him it is 8:30 p.m. Personally, my brain can hardly be called functional at 8:30. So thank you so much for being with us virtually today. And without further ado, please welcome Dr. Jamie Hall for his talk today titled Problems with Plasmids.
Great, thanks very much, Chava. So I'm doing, when I've been doing these talks, I'm just trying to get a feel for what the audience is like, because I don't really know a lot about what you all do. So I'm just going to launch a poll. So you can click and choose, and that might help me work out what areas I need to be slower or faster on to help you follow what's going on. Okay, another couple of seconds. Oh, everybody. All right, so there are the results. So we're really even spread here. Great. That speaks to a multidisciplinary institute, I think. Great. Okay. So like Chava said, I'm Jamie Hall. I'll do my acknowledgements and everything at the start. So I'm talking about problems with plasmids. This work has been done at the Universities of Liverpool, Sheffield, and York. And partly it's sort of the continuation of some of the postdoctoral work I did in Mike Brockhurst's lab. And Mike's actually now moved to Manchester. So we're all across the north of England here. And this work's been funded particularly by NERC and the ERC. Okay, so I'm really conscious giving this talk that I'm speaking to people whose work has been absolutely formative in my understanding of plasmid evolution. But as I wasn't entirely sure of what the full composition of the audience would be, I hope you'll forgive me if I have a bit of a gentle introduction, and particularly, Eva, I'm going to have a slide telling you what a plasmid is. I'm really sorry. I'm really sorry. Okay, so from the food that we eat to the environment that we live in to our own bodies, bacteria define the world we live in, and they underpin our health, and they underpin our disease, and they underpin the health of our environment as well. But one of the really amazing things about microbiology, it was only really very recently in human history that we really got an understanding of what microbes actually were, that we really got to see them up close. It was only when people like Leeuwenhoek and Hook developed, started grinding bits of glass and looking down them, that we looked down our microscopes and we suddenly realized that biodiversity and life on Earth went so much deeper than we previously thought. You can't see the slides. That's because I'm not sharing my screen. Hold on. Right, sorry. Start again. Okay. So we didn't know about microbes until relatively recently in our history. And it was only when we turned our microscopes on them that we really got a proper understanding of how deep biodiversity on this planet went. So recent years, we've seen a massive acceleration in our understanding of bacteria, thanks to the availability and the cost of genome sequencing. Devices like the Illumina HiSeq and the Oxford Nanopore are effectively acting like a new kind of microscope. They're letting us see microbial communities in ways that we could not see them before. So we look down our microscopes now and we see all this kind of mess of data. And of course, this requires a bit more interpretation, but the real picture that I think is emerging, not just from microbial genome sequencing, but I think one of the really biggest pictures from genome biology in general is that bacteria evolve quickly to cope with environmental challenges. And they do this because they exchange genes with one another. So if we look down the microscope, we look at our microbial community, if we sequence it, we realize that actually they're evolving in a way that's quite unlike animals and plants. So this is horizontal gene transfer. This is when organisms acquire genetic information from individuals that are not their parents. And the key thing about horizontal gene transfer that makes it so fascinating is that it's facilitated mainly by mobile genetic elements. So in this case, it looks like there's a plasmid being exchanged. Of course, mobile genetic elements themselves are very diverse. And these mobile genetic elements can have lives of their own, essentially. These are a different participant in this process. If you think of horizontal gene transfer as an exchange of information, it's unlike the exchange of information that's going on as I'm talking to you, in that there's another player in this dynamic, and these are the mobile genetic elements. So we should all be interested in horizontal gene transfer because it underpins many of the microbial traits that we're interested in terms of producing our food, causing our disease, and keeping our environments, maintaining our ecosystems. And so I believe that to understand bacterial evolution in general, and so the emergence and horizontal gene transfer in particular, we really need to understand the behavior and evolution of these elements. Okay, so I'm going to talk about plasmids. Plasmids are really important in horizontal gene transfer because it's thought that plasmids are responsible for most horizontal gene transfer that we find. Plasmids are bits of double-stranded DNA that replicate separately from the chromosome in a bacterial host. And they're interesting because they are really widespread. So they're found in all different types of genera of bacteria. They're naturally occurring, so I know lots of people are familiar with plasmids because of their role in biotechnology, but it's really important to emphasize that any kind of environment that you go to, you can find diverse mobile genetic elements. They're these important vehicles for gene exchange, and the key thing is that because they have this autonomy, because they replicate separately in a bacterium, they are Darwinian entities. They have fitness interests, which may be different from the other genes in this bacterium. Okay, so we want to try and understand plasmids. So the system that I've mainly used to understand plasmids so far is this one. So this is a system of plasmids and a bacterial host, and they were isolated from a field of sugar beet in Oxfordshire about 30 years ago. And the nice thing about this system is we've got a set of different types of plasmids, which are mercury-resistant plasmids, which were found in this environment. And I just want to emphasize the size of these plasmids. So these are all what we call megaplasmids, and that they are hundreds of kb in size. So they're relatively large for plasmids. And that means that they carry lots of genes. Interestingly, we don't know what many of those genes do. But one thing we do know about them is that these plasmids all encode mercury resistance. Now, these plasmids are all got this kind of name, Pqbr, and a number. It can be quite difficult to track those numbers if you're reading them a lot, particularly if you're not familiar with them. They all have individual lives of their own. When I see them, they're like my little babies. But in this case, I'm gonna sort of help out a bit by trying to keep these colors constant through the presentation, just so you can track the blue one, the green one, and the yellow one. Okay. The host that I work with in this kind of situation is a naturally occurring bacterium which exists, which came from the exact same site as the plasmids came from, but it's originally plasmid free. So this means that when these plasmids go into this host, this is a new association, but it's a natural association. It's one that we might expect to occur in the wild. Okay. So we can take these plasmids and we can see the fact that they have these different fitness interests from their host, because what we can see is when these plasmids, one way that these fitness differences manifest is that when we take these plasmids and we put them into a test strain and we compete them against a reference plasmid-free strain of bacteria, we can observe fitness costs. So the way this experiment works is we take each of these plasmids, we put them one at a time into a test strain. If we calculate relative fitness, if this is above 1, then there's a benefit to having the plasmid. If it's below 1, then there's a cost to carrying this particular plasmid. And what we can see is when we do this in lab media, if we do this in lab media, we see that all of these plasmids have a fitness cost in that this relative fitness value comes out below 1. But we can see that these costs can be outweighed by the benefits. In this case, the benefits that these plasmids provide in encoding mercury resistance or mercury detoxification. As we add mercury to the environment, these costs get outweighed by selection, and there becomes a fitness benefit to the plasmid. And this pattern is also seen in potting soil. We can do the same experiments in a natural kind of environment, like where these plasmids, these bacteria are isolated from. Again, fitness costs when there's no mercury around, fitness benefits when there is mercury around. So plasmids can provide useful traits, so they're beneficial under these kinds of situations, but these situations are actually pretty rare. If we look at the concentration of mercury in the soil in the UK, for example, it's much, much lower. And actually, these plasmids, these mercury resistant plasmids, were all isolated from pristine agricultural fields. They weren't polluted, mercury polluted regions. So actually the concentration of mercury in the environment where these plasmids was found was all pretty low. What this means is that under these natural conditions, these plasmids have a fitness cost. And this phenomenon of plasmids causing fitness cost is actually really common. So we can look at Pseudomonas aeruginosa and a bunch of different plasmids, and we can see that these plasmids are costly. We can look at Klebsiella pneumoniae, which is a pathogen in an opportunistic pathogen. We can put these plasmids in it, and we can see that this one also has these costs. We can see again E. coli and this other plasmid. In all of these cases, there are costs, but again, in all of these cases, if we select for the plasmid, we can outweigh these costs and provide a fitness benefit. But the reason why we still need to understand where these fitness costs come from is that these beneficial genes on the plasmid, under an environment where they're beneficial, they can actually recombine onto the chromosome, and then you end up with a redundant, costly plasmid that we still need to explain. And this is definitely the case for these Pqbr plasmids, because in these cases, the mercury resistance genes are actually on a transposon, and we've seen in the lab many times these resistance genes jump off onto the chromosome. So to understand plasmids, to understand the distribution of plasmids, and hence to understand horizontal gene transfer of the traits we're interested in, we have to understand plasmid fitness costs. We have to understand how bacteria and plasmids conflict with one another and how they might get along. Okay, so I've got another poll here. This is based on, so like I say, we can measure these fitness costs when a plasmid goes into a bacterium. And there are many different ways that we might imagine a plasmid to cause fitness costs, right? So in our case, we have these megaplasmids, and they present more DNA to copy, and that might be costly. They represent more genes to express, and that might be costly. They might interact either with other mobile genetic elements, other plasmids or chromosomal islands and so on. So there might be clashes with these systems. There might be clashes with the general cellular network and cellular physiology. And it may be that plasmids are costly because they encode toxic products. So I'm going to activate the next poll if I can. And I want you to vote on where you think the dominant source of plasmid costs comes from. And almost everyone's voted now, so I'll shut the poll in a minute. But I'll say when I started this project, I was thinking these are megaplasmids, right? They're actually 5% or between 5 and 10%. They represent between 5 and 10% of the DNA in these bacteria. And so when I started this project, I was like, right, this is going to be costly because of this extra 5% of DNA being added. It's more DNA to copy and more genes to be expressed. And most people agreed with me when I started this project that the main source of plasmid cost was going to come from these kind of generic factors associated with plasmid size. Big plasmids means more proteins to be made and more DNA to copy. And I'm going to share some, I can share these are the results from the poll. And I'm going to share my scientific results, and we can see how well this compares. Okay. So a key thing in this that has been shown by many people is that these costs can actually be resolved by evolution. So a number of studies over the years have shown that mutations occurring to the plasmid or to the chromosome can effectively resolve the fitness costs of plasmid carriage. And in our case, we've got this example. I'm actually just going to tell you what you're going to see before I show you. So there's an example that I showed of how plasmid fitness costs can be resolved by evolution. This is with this plasmid Pqbr55. It goes into the bacteria and it imposes a fitness cost such that the colonies which are formed are actually really small. What I did was take a video of these colonies over a 48-hour period to see what happened with these colonies. And what you can see, so these are the colonies, they're carrying a gene which causes them to appear blue. And what you can see is that over this 48-hour period, we see these breakout mutants, which effectively have ameliorated, they still carry the plasmid, but they no longer form this small colony phenotype. And you can see actually in some cases a single colony produces multiple different of these breakout mutations at once. So this shows that this process of compensatory evolution, where mutations resolve the fitness cost of plasmid carriage, occur really rapidly, really readily, and really reproducibly. So a number of studies in our group looking at these particular Pqbr plasmids in this bacteria have now identified a number of different compensatory mutations which have appeared in evolution experiments like the one that I've just shown you. And the three ones I'm going to talk about today are this system, so the GAC-AS system, which is a two-component signaling system, and essentially this is a global regulator of secondary metabolism. And we've seen both of these plasmids, if you put them into a bacterium, you allow them to evolve, they get mutations in this global regulator of secondary metabolism. This gene's on the chromosome. We have another chromosomal gene called PFLU4242. We don't know what it is, we don't know what it's doing, but this one repeatedly appears in our plasmid evolution experiments with three different plasmids from our Pqbr plasmid collection. The final mutation I'm going to talk about is this one, Pqbr5759. This one is different from the other two because this one's actually on one of the plasmids, on Pqbr57. It appeared in about 25% of populations that I evolved with this plasmid through different types of mutagenic process. And this gene is predicted to be a lambda repressor. So these are the mutations that appeared in previous experiments. And so then what we can do is we can generate mutants in the lab with specific mutations in these genes and see how they affect the fitness cost of the plasmids. I'm just going to focus on these two plasmids, Pqbr57 and Pqbr103. The reason for that is mutations arise so quickly for this Pqbr55 plasmid that it's very difficult to do the controlled experiment. Okay, so this work has been pre-printed on bioRxiv, so you can track it down afterwards. I kind of give away the ending here, so I'm going to skip over dwelling on that too long. So here's the experiment. We either take no plasmid, we take Pqbr103, or we take Pqbr57. We put them in each of these strains, either the wild type or strains lacking GAC-S or PFLU4242, and then we compete them against the wild type competitor. Again, fitness below 1 is a fitness cost. So if there's no plasmid around, if you knock out GAC-S or you knock out PFLU4242, there's actually no fitness cost when we measure this in Kb broth in the lab. If we take this plasmid Pqbr57 in a wild type bacteria, this plasmid has a fitness cost of 17%. But these mutations resolve the fitness cost, and particularly PFLU4242 is very effective at resolving this fitness cost such that we can't detect a fitness cost in strains which have this mutation. A similar but perhaps more pronounced pattern is seen with Pqbr103, the larger megaplasmid, in that this one has a 51% fitness cost in the wild type background. Knocking out either of these genes effectively resolves the fitness cost. So these compensatory mutations ameliorate these two very different plasmids. Okay, we can look at the mutation on the plasmid as well. So from our evolution experiment, we got two different mutants, or I should say one evolved plasmid which had no mutations and one evolved plasmid which had a single base mutation across its 307 kb plasmid. This single mutation caused a single amino acid change in this putative lambda repressor. So we can take either of these plasmids, we conjugate them into an ancestral strain, and then we compete it against the wild type strain. And again, we see the ancestral variant of the plasmid has a fitness cost. This single base pair mutation on the plasmid effectively resolves that fitness cost, ameliorating Pqbr57. So we've got these two different principal mechanisms for compensation, and in an evolution experiment I did, it appeared that they were mutually exclusive, such that you only need one of these mutations and it does the job. And this was supported by experiments that show that there's no added benefit of having both the mutation on the chromosome and the mutation on the plasmid. If you knock out this gene on the chromosome, you resolve the fitness cost. If you have a single base pair mutation on the plasmid, you resolve the fitness cost. And if you have both the knockout and the single base pair, the mutation on the, it's the same. You don't get an additional boost. So what this tells us is that one of these, all of these genes are important. You break one of them, and the whole fitness cost problem is resolved. Another little bit of data here. So we might think that if we compensate the cost of the plasmid, it might affect the way the plasmid behaves in terms of the ability of the plasmid to undergo horizontal gene transfer. This doesn't seem to be the case. So the wild type plasmid conjugates effectively into Pseudomonas fluorescens. It conjugates less effectively between species, but it still conjugates effectively. These mutations that ameliorate the cost of the plasmid do not have a trade-off in terms of the ability of the plasmid to transmit horizontally. Okay. So this allows us to go back to our little thing here, and I've shown that these bacteria still carry the plasmid, so they're still having to copy the DNA, and yet they've resolved the fitness cost. So I think we can say that it's not the fact that they're having to replicate DNA which is causing the plasmid fitness cost. We can also rule out somewhat that it's conjugation that's causing these fitness costs, because like I showed, we can ameliorate the cost of the plasmid, but they still conjugate. Okay, so we've got a kind of complicated situation going on, right? Either of these plasmids has some kind of interaction that involves these three genes, the GAC-AS system, this gene PFLE4242, this gene on the plasmid. There's some interaction between all of them such that if you break one of them, you resolve the fitness costs, right? There's some kind of cascade that's going on between these things, or subsequently, which is causing the fitness costs. If you break one of these things, then we solve the fitness costs. And we wanted to understand what was going on there. We wanted to understand what these question marks were, basically. And so to do that, we did an RNA-seq experiment where we either had bacteria with no plasmid or either of these two plasmids. We have either of these situations of amelioration, either where they have no amelioration, we delete the GAC-S, we delete the 4242, we've got this single base pair mutation on the plasmid, 3 replicates of each, and then we did RNA-seq to look at differential expression. So what I'm first showing you is gene expression from the chromosome and from the plasmid, both the Pqbr57 on the left, Pqbr103 on the right, under these different conditions of amelioration. And what you can see is that there's no global effect of, there's no global effect on transcription of these mutations. It's not the case that we get a dramatic downregulation of all plasmid genes, or a big bulk downregulation of plasmid genes that is associated with amelioration. And so this makes me think that it's not the case that there's more gene expression that is causing plasmid fitness costs. So what are the responses to plasmid acquisition then? So what I'm showing here is a heat map. Each column is a differentially expressed gene, either following Pqbr57 acquisition or following Pqbr103 acquisition. I've grouped them according to their response to the different plasmids, whether they're upregulated in red or whether they're downregulated in blue, in both plasmids, in one plasmid, in the other plasmid, in opposite conditions, and so on. So these are the differentially expressed genes, more than twofold differentially expressed, and the ones which are boxed in have a false discovery rate of less than 0.05. Okay, so we can see that there are these common responses to plasmid acquisition, and then there are some divergent responses as well. And by looking at the genes which are involved in this, we can get a feel for what the common and divergent responses are. And basically, it pans out like this, that the genes which are upregulated by both of the plasmids are overrepresented in SOS response and signal transduction. So the SOS response is a response of bacteria to DNA damage. There are also these sort of divergently expressed genes which were overrepresented in metabolic pathways, particularly nitrogen compound. But interestingly to me, almost all of the genes which were upregulated by both of the plasmids were associated with chromosomal mobile genetic elements. So these are prophages, SOS response regulated genes, and a chromosomal transposon TN6291. Okay, so these are genes which get upregulated by the plasmid, but we don't know whether these are cause of the fitness cost or an effect of the fitness cost. It's very difficult to disentangle these things with RNA-seq data alone. So to understand this, I picked a selection of these genes and I cloned them into an expression vector to see whether expressing them would have some kind of fitness cost. Okay, activation of genetic elements. Okay, so what we can see here at the bottom is these are the genes that I expressed. We're showing what happens if they're induced in red. And basically when the red line is below the blue line, there's a fitness cost of expressing that gene. The key one I want to draw your attention to is this one here, PFLU 1169. This gene is associated with this prophage 1 locus, and it encodes the holin, which basically makes holes in the cell membrane to allow the phage particles to be released. But in an interesting twist to this story, this isn't actually a full prophage. This is what we call a tailosin. So it doesn't actually encode the packaging part of the phage, packaged DNA. It's essentially just a phage tail. And these are well described in bacterial genomes as being essentially bacteriocins. These are molecules which are released by bacteria to kill their competitors in competitive situations. And this is kind of interesting to me because basically what we see is we get a bacterium. So I'll rewind. Expressing these genes is costly. Producing these bacteriocins is costly because it requires a cell to be lysed to release these toxins. It's essentially a spiteful act because the individual cell dies to remove a competitor to help its kin. Right? That's the kind of evolutionary dynamic that's going on here. Now, the idea that bacteria upregulate these particular systems when they acquire new genetic information, I think is kind of interesting because it suggests that this might actually be a specific reaction to the presence of competitors, such that when they get new genetic information in the form of a plasmid, they release these bacteriocins to kill their competitors. Okay. So this is the slide I just showed you. These are the fitness costs of the plasmid. These are the differentially expressed genes associated with plasmid acquisition. What happens when we get the mutations that compensate those fitness costs? So what I'm showing here is what's happening to these genes, either when we get the PFLE4242 mutation or when there's a single base pair mutation on the plasmid. That's these different groups here. And then when there's no plasmid, when there's Pqbr57 and when there's Pqbr103. So the first thing I want to draw your attention to is this line where there is no plasmid and we get the PFLE4242 mutation. What you can see is that there are no differentially expressed genes associated with just knocking out this gene. It has no effect on transcription at all, which is really interesting because it chimes with the fact it has no effect on fitness if we just knock it out by itself. But if we knock this gene out in concert with one of these plasmids, we see all of these effects or all these differentially expressed genes which changed gene expression following plasmid acquisition. They all go back essentially to normal. Almost all of them come back down to the baseline, which again is consistent with what we found phenotypically in that fitness costs were resolved. And we see a similar pattern happening with a single base pair mutation occurring on the plasmid. Okay, I'm not showing the GAC-S effects on here. The reason for that is that because GAC-S is a master regulator of secondary metabolism, if you just knock out this gene, you get huge amounts of differential gene expression. But I'm just going to focus on one particular effect of knocking out GAC-S, and that is if we knock out this gene GAC-S, we decrease the expression of this other gene involved in plasmid fitness cost, PFLE4242. What this means is that the GAC-AS system is upregulating PFLE4242. So knocking out the GAC-AS system basically helps resolve the cost of plasmid carriage because we're removing this other element which is important for plasmid fitness cost. Okay, so this was a complicated picture I showed you before. We can start filling in some of these gaps. So the SOS response, which is activated by DNA breaks, activates this prophage 1, particularly this gene which encodes this holin, makes holes in the membrane, cell damage, that causes fitness costs. This may not be the only way in which the SOS response causes these fitness costs, so there may be other effects as well, but this is a confirmed pathway. We see this particular gene is upregulated by the plasmids. If we express this gene by itself, it causes similar fitness costs. Up at the top here, it's a little bit confusing, but what we can do is we can show that actually a parsimonious explanation here is that mutating GAC-AS, this master regulator of secondary metabolism, actually exerts its effects by reducing expression of PFLU4242. Essentially, there are two ways of doing the same thing. It's cutting off your hand or cutting off your arm. Cutting off your arm ends up being a lot more destructive than a mutation to this downstream gene of this much more diverse response. And we can also see in the RNA-seq data that this multifaceted response affects a bunch of secondary metabolic pathways, which we could imagine would have some kind of trade-off in a natural environment. Okay, so we solved some of this story, but what's going on here with this plasmid-borne gene, this one that we mutated? How is this one interacting with this system? So this gene is disruptive. If you have the wild type variant, we have a fitness cost. If we have a single base pair mutant in it, we resolve that fitness cost. How does that work? So there are two different hypotheses. Either this gene itself is toxic and it's directly interfering with the cell, or it's interacting with other plasmid genes to cause a fitness cost. To investigate this, we can take the wild type variant of this gene and we can clone it onto an expression vector. And we can also take the mutant variant and put it on an expression vector as a control. If this gene is toxic and it directly interferes with the cell, then putting it on this expression vector will be sufficient to recapitulate those fitness costs. Whereas if it requires other plasmid genes, we can put in the rest of the megaplasmid that is otherwise doesn't have a cost because it's got the mutation in the gene, and we can see basically whether we can complement those costs like this. Okay, so what do we see? So this is just our control. If we have the wild type megaplasmid, we get fitness costs, regardless of whether we have an expression vector with no insert, an expression vector with the wild type variant, or expression vector with a single base pair mutation. If we have the plasmid with the mutation in it, that one, of course, doesn't have a cost by itself. If we put in the expression vector with no insert, no cost. If we put in the expression vector with the wild type variant, we can complement it and we can actually see the cost coming back. And if we don't have this megaplasmid, then we don't get a fitness cost, even from the wild type variant of this protein. So what this says is that we're in this situation where there's some kind of interaction with other plasmid genes, which means that this protein causes a fitness cost. Okay, so what does this gene do? We do some predictions, and it says that it's a repressor-like regulator, and we think it's interacting with other plasmid genes. So if it's a repressor and it's repressing some plasmid genes, if we break it, we should be able to see that effect in our RNA-seq data. So what I'm showing here is what happens to expression of genes from the plasmid Pqbr57 when it becomes ameliorated, either by PFLE4242 mutation or by the single base pair mutation on the plasmid to this repressor. And what you can see is that, again, there are some common responses to amelioration. There are some divergent responses to amelioration, but the key thing are these two genes here, which are specifically upregulated by this single base pair mutation in the repressor, and they're not affected by this other mode of compensatory evolution. And interestingly, these are the two genes on the plasmid that encode the putative partitioning machinery of this megaplasmid. Now, these genes are consecutive, they form an operon. And if we look upstream of this operon, what was really cool is that we see repeat regions such that this repressor may be able to bind to these regions. And so if we break the repressor, then we get more expression of these genes, which is what we see in the RNA-seq data. But if I looked back in my evolution experiment, where we originally found the mutations in this repressor, we also found mutations that affected this upstream region. So mutations either to where this repressor binds or to the repressor were both found in the evolution experiment, suggesting that it's this particular interaction between the repressor and this region upstream of the PAR operon that causes repression of the PAR operon, that is what is responsible for the fitness costs. Okay, so the partition genes, for those that don't know, essentially these megaplasmids are so big that they've got machinery to make sure that they get one plasmid in each of the daughter cells. It mechanically distributes the two copies of the plasmid to the daughter cells before they divide. So is it really the case that upregulating the partition genes reduces plasmid fitness costs? Well, to test this, we do the same experiment where we take the partition genes, we put them onto an expression vector, and then we see whether they ameliorate the fitness cost of the plasmid. What you can see here is if you don't have a plasmid, expressing these PAR genes doesn't have a particularly significant effect. If you have the plasmid Pqbr57 with no insert or no expression vector, there's a fitness cost. But if that expression vector has the partition genes and is overexpressing the partition genes, we ameliorate the cost of Pqbr57. But interestingly, if we express those same PAR genes that ameliorate one megaplasmid, but we try it with the other megaplasmid, it actually has the opposite effect. And that fitness effect is actually exacerbated if we express those partition genes with the heterologous plasmid. Okay. So we can fill in this little bit of data here now, where Pqbr5759 inhibits the partition genes, which would otherwise be inhibiting this negative interaction between this plasmid and the PFLE4242 genes. Okay, one final story from these data, which I thought was really interesting. So, sorry, before I go to that, I'll expand on this, what the implications of this is. So the cool thing about this is that the plasmid actually holds the proteins it needs to ameliorate itself. It's just repressing them. And this is kind of cool because actually other plasmids have been found that have proteins which can reduce their fitness costs. There are these silencing genes carried by plasmids which silent plasmid expression to reduce their burden on the host and to reduce their plasmid fitness costs. In this case, it's cool because we've actually seen this function evolving during our evolution experiment. Okay, so I got away with myself before. The final cool story from this data is, what are these genes here that remain upregulated? Right, there are these three genes that remain upregulated when you get amelioration by knocking out PFLE4242, when you get amelioration by knocking out Pqbr5759, they're still upregulated in the GAC-S mutant as well. Turns out that these three genes are all co-located. They all are part of this transposon TN6291, and they all actually encode the transposase for this transposon. They remain upregulated when they have the megaplasmid, even though the main physiological disruptions are carrying these plasmids is now ameliorated. So what this means is that these plasmids actually can upregulate this transposase independently of all these other fitness costs that they're causing. And that can cause transposon mobilization potentially. And we actually see these plasmids in an evolution experiment that we did with Pqbr57, we actually see this transposon becoming activated during the experiment, jumping onto the plasmid and using the plasmid as a vehicle to transmit into new species. This is a chromosomal mobile genetic element. It doesn't have the ability to transmit itself normally. Here we see it using a new vehicle that has arrived, and it's directly responding to the presence of that vehicle by upregulating the genes that it requires to get in and go, basically. So this is basically the equivalent of getting the ping saying that their taxi has arrived and that they can now use that to move into new backgrounds. Okay. So what causes plasmid fitness costs? Hopefully I've convinced you that the majority of plasmid fitness costs are actually due to specific gene interactions both with other mobile genetic elements and with other chromosomal gene products. Now, I'm not saying that plasmids don't have a metabolic cost. We can calculate the metabolic cost. We can actually see it in some cases. What I'm saying is that in natural settings, these costs are dwarfed by the specific genetic conflicts that exist between plasmids and chromosomes. And actually, those metabolic costs may prove to be negligible in natural settings in the context of rampant horizontal gene transfer, in adaptation to other selective pressures, and so on. What this means is that to predict these fitness costs, and thus to understand bacterial genome evolution in general, we need to start looking at particular gene co-associations. What genes appear together, what genes don't like one another, what genes do like one another, rather than looking at generic proxies like size or codon usage. Okay, so this is my kind of summary. Specific gene interactions responsible for fitness costs. And this really comes about by this observation that single base pair mutations can really sort out the cost of even these really big plasmids that increase genome size by 5%. Okay, I've shown you that mobile genetic elements are in conversation with one another. They're detecting one another and they're responding to one another to enhance their own fitness interests. And I've also shown that we have these two different modes of plasmid amelioration, a mutation on the plasmid and a mutation on the chromosome. And these can actually, although they have the overall same effect in terms of reducing plasmid fitness cost, and we see both of them emerging in our evolution experiments, the consequences of these things for genome evolution and for microbiomes more general could be quite interesting. This mutation on the chromosome ameliorates multiple different plasmids. So you might end up with a bacterium becoming essentially a source of plasmids for its neighbors. On the other hand, this plasmid mutation just ameliorated the cost for itself, but it becomes transmitted when that plasmid then moves into different backgrounds. So the key message is it's important how plasmids are ameliorated, and there are consequences of this. Okay, so what should we do next on this project? Should we work out the mechanism a bit more about how these genes cause fitness costs? Should we try and understand a bit more about how mobile genetic elements interact with one another and how this affects ecology and gene transfer? Should we investigate this beyond this particular bacterium and see what happens in different bacteria or in different environments? Or should we investigate this question of whether mutations on the chromosome or the plasmid are more important in different settings? So I'm going to launch another poll. And you can tell me what I should be doing next. Okay, I'll give you 5 more seconds. 3, 2, 1. Great. Okay. So I share the results. So the most popular one was to expand the scope to other species and environments. So all of these things are actually happening. So I've got my first PhD student, Tanya Horn. She's going to be delving into what this gene of unknown function, PFLU4242, is actually doing and how it causes these conflicts. Rosanna Wright, who's a postdoc who's carried on a project with Mike, who's over in Manchester. She's been following up this question of different routes to compensation. I just submitted, well, it was in January, I got the QC back in an email today saying that these are going on the sequencer soon. So I've got RNA from the plasmid mutant in 5 different species of Pseudomonas. And I've got a new PhD student starting in the autumn who's going to be looking at this question of conversation between mobile genetic elements. So I'm really glad that you all agree that all of these things are worth following up. Okay, so I've been talking about megaplasmids in soil and this particular strain of soil Pseudomonas. So this has been a little bit abstract and looking at these particular gene conflicts. What's the bigger picture here and why should we care? Or if you are somebody who's very, very interested in how microbiology helps human health, what can I offer you? So I've been talking about soil Pseudomonas, but of course Pseudomonas itself is an actually very important medical concern. Pseudomonas are ubiquitous. They're found in all different kinds of environments. They're really diverse. They're really metabolically versatile. But they're also responsible for a lot of disease, and resistant Pseudomonas are they're a major threat. They're opportunistic pathogens And this threat is predicted only to get worse with the acceleration of antimicrobial resistance. So a paper that I worked on last year with Adrian Cazares, who's now in Cambridge, did some work looking at a megaplasmid family that he had initially identified in some isolates from a clinic in Thailand, which were driving dissemination of multi-drug resistance in Pseudomonas. And this was really made possible by the Oxford nanopore sequencing that enabled us to, or enabled Adrian really, to see these megaplasmids in genomes which otherwise would have been really fragmented with a short read sequencing. And once he pulled out those megaplasmids with these long reads, he then went back to databases and said, okay, amongst the short read databases, how common are these megaplasmid families? So I'm going to show that data in just a minute, but the really worrying thing about these plasmids is that they're big, which means they carry lots of genes, and particularly they carry lots of resistance genes. This whole variable region of this plasmid is just chock-a-block with resistances to aminoglycosides, beta-lactams, fluoroquinolones, and so on. These plasmids are cosmopolitan, so they've been found in clinical isolates from around the world, but also interestingly, related megaplasmids of this family have been found both in healthcare settings and environmental settings. And this has been enabled by this kind of research where, going back through the short read database, Adrian was able to find that relatives to this plasmid were found in 1 to 2% of all Pseudomonas genomes, which might not seem like super high, but if we think this is all Pseudomonas genomes, which are heavily biased towards species which are known not to carry so many plasmids, that actually this megaplasmid family is much more widely distributed than anybody previously thought. So this is a diagram showing three different megaplasmids from this family. The two at the top are megaplasmids from clinical Pseudomonas aeruginosa. The one at the bottom is from a soil strain which doesn't carry any antibiotic resistance genes. And what's interesting is that we really see this strong conservation of the backbone of these plasmids, but a region of antimicrobial, or a variable region, which really speaks to the ability of plasmids to provide environmentally or locally adaptive genes. In these clinical strains, they're providing antimicrobial resistance. In the environment, they're providing metal resistance and metabolism. And one reason I was really interested in these plasmids, that I'd already had my eye on some of these plasmids, because I identified that these are actually kind of distant relatives of the two plasmids that I focused on in this talk, speaking to a diverse family of megaplasmids, which are kind of all around us really, but are potentially accelerating this really recent problem of antimicrobial resistance. So I took two of these plasmids, this clinical one from POZ176 at the top, this one from soil in the middle, and then of course the two Pqbr plasmids at the bottom. And I measured their fitness costs and their conjugation rates. And what we can see is that despite this clinical plasmid, particularly POZ176, which encodes loads of resistance genes, despite the fact that it's half a megabase in size, it has no detectable fitness cost in our Pseudomonas fluorescent SPW25 strain. And on top of that, it also has a very high conjugation rate. So up to 50% of the recipients, if you culture donors and recipients for 48 hours, up to 50% of those recipients had the plasmid by the end of the experiment. So what this shows is that these megaplasmid families are spreading multi-resistance, probably because they do not necessarily, even though they're big, they don't necessarily have a big cost. And even though they're big and carry lots of genes, it doesn't mean that they're not jumping around between different strains, between different species, in our soil, in our sinks, in our hospitals. Okay, so I want to understand how these plasmids are affecting the phenotype of the bacteria that acquire them. I wanted to understand what the features are of these strains that enable them to maintain them and transmit them. And I want to understand how these megaplasmids are spreading in multi-species communities. And this is the subject, or some of the ambitions I want to try and get started on some of these questions in my first grant, which I got last year from the Academy of Medical Sciences Springboard Award. And Sam Riti is a new postdoc on this project who's been doing a really great job getting the experimental side of stuff sorted out with this. We've got some of these plasmids labeled with fluorescence genes so that we can actually track their maintenance and transfer now in communities and in individual strains. Okay. So these are the take-home messages. If we want to understand bacterial evolution, we have to understand plasmid biology. And if we want to do this, we have to do experiments, right? We can't get away just by sequencing everything we come across. We have to actually do some experiments to understand the causal drivers of this process. And with that, I'll be very glad to take any questions. Before I go, I'm just going to plug a new initiative. This is a comic that I've written about plasmid biology. Well, it's about microbial diversity, microbial evolution, and the creativity and imagination that we all need to have as scientists, and basically how exciting microbiology is. So I'd encourage you, if you're interested, to visit the website. But otherwise, thank you very much for listening. And thank you very much for having me. It's been really fun. So I don't know if anybody has any questions. I'll stop the share so that.
Hey, Jamie, I have a question.
Hi, Devo.
How are you? We'll talk after. Well, I was like, I found it was interesting that you saw the SOS, response that is overactivity when the plasmid arrive into the cell, and I was wondering if you looked at that work that was from Didier Mazel, where they found that when the plasmid transfer into like a bacteria, like the single strand DNA is what's activate the SOS response, and that can be maybe like producing, like by activating the SOS response to activate the expression of a bunch of other genes, and maybe also like exacerbate the cost of the plasmid. Is it something that you've been looking at or interesting?
Yeah, that's a really interesting question. So yes, the SOS response is triggered when plasmids are required because of the single-stranded DNA. We don't think that's the case here because we conjugate the plasmids in. So if you remember that work by Didier, he shows that this SOS response is activated when the plasmids go in.
Yeah.
And then when they actually become double-stranded DNA sitting there, then the SOS response is turned off. Mm-hmm. Whereas in this case, we see this fitness cost from generation to generation, right? We grow a colony, we pick it, we restreak it, we do these fitness experiments on it. We can still keep measuring that fitness cost. The strains that I did for the RNA-seq experiment, they were new transconjugates, but I plate them out, I restreak them, I grow them in media. So that's generations and generations since they first acquired that plasmid and were exposed to that single-stranded DNA, which is why I think this is double-stranded DNA breaks being generated as some downstream effect of the plasmid, something that's going on with that.
Okay.
Jamie, I had a question about the PAR genes on the plasmid. So can you walk us through that again? So somehow that the mutation on the plasmid downregulated the PAR genes?
Right. So the gene on the plasmid that got mutated is a repressor. So if you break a repressor, then you get stuff turning on. Yeah. And so what we see is we break the repressor, the PAR genes turn on, and the plasmid cost goes down.
So how do you explain that link? How does a upregulation of PAR genes decrease the cost of the plasmid? Yeah,
so that's really interesting. And again, we see that cost going down, but we only see it going down for one of the plasmids and not for the other one. So it's a specific interaction with something else that's going on in the plasmid. So we know that. And we also know that people have worked on these PAR gene families for a long time because they've got homologs on the chromosome, and they've got homologs in eukaryotes as well. And they come in two parts. So there's a PAR A and there's a PAR B. The PAR B binds the DNA, and actually it has silencing effects by binding DNA, and it also may potentially protect DNA. So it binds to DNA, and you can see like DNA's fingerprinting, you get protection of that region. And so we don't exactly know how it's happening yet, but I'm conceptually linking it with that double-stranded DNA break problem. So perhaps what's happening is you upregulate the PAR genes, and somehow they are helping to prevent double-stranded DNA breaks, which otherwise would activate the SOS response. But this is just me hand-waving a bit, and I'd be really interested to speak to some people who know a lot more about that system to kind of, I mean, I've chatted with a couple of people and they say it sounds reasonable to say, well, PAR gene binds DNA and it can silence and it can protect, but I don't know whether there are any experiments we can do to actually understand that in more detail.
I see a question from Liv.
Yeah, do you want to ask it, Liv, or do you want me to read it?
Since Java is not moderating, I'm starting.
Hey, Jamie. Yeah, so my question was about your RNA-seq data where you saw that the prophages became upregulated and that's like stayed on. Have you done any tests to see if the toxins are active or done a spotting test, anything like that?
Not yet. The reason for that is that these toxins are very specific. So they, you can sample a whole bunch of different strains and it will target some of them and not others. And so what we don't know is what target to use for them that we'll be able to actually find something that's susceptible. I'm sort of in the process of testing a knockout of that cluster, and potentially if you knock it out, it might become susceptible to it. So that could be some way of testing it. I tried to get a strain from Dave Baltras that he said was susceptible, but it turned out that it wasn't. So yes, that's definitely something I want to do. And that would allow me to test this question of whether these having these functional toxins actually have some adaptive benefits when you acquire a plasmid. So if you acquire a plasmid, does it help to upregulate these in a competitive situation?
Thanks.
Hey, Jamie.
Hi, Chris. Good to see you.
Hey, likewise. Though it was more fun to see you in bars in Aberdeen at a conference, like you said, back in the good old days.
Back in the good old days.
But yeah, great talk. I'm curious, to some extent, I guess, why, if these compensatory mutations sometimes are so easy to arise, why weren't they already there, right? Is it just either the environment or the host is naive, and so there just wasn't any prior selection to deal with the problem? Or do you think that there were, in some other context, there's fitness trade-offs, kind of similar to like phage resistance? Often, phage resistance mutations arise easily, but they hurt the fitness of the host because they like reduce nutrient uptake or something like that. And so you showed one example at the end of a massive plasmid that wasn't too costly. So maybe it has, already dealt with that, at least for what was tested. But yeah, kind of why? If they're so easy, why aren't they already? Yeah,
I mean, I think that's a great question. So one possibility is that this could just, every time we pick a bacterium out of the environment, we're basically taking a snapshot, right, of a genome which is in constant flux. So if it's not sorted out a problem yet, that's not really surprising because it's something which is constantly gaining genes, losing genes. It's part of a dynamic system. So, but I think I've got a better answer, which is that I do think there's a trade-off. And you almost took the words out of my mouth when you said phage resistance, because I've been like scouring the literature to try and work out what this protein could be doing, because all it seems to do is cause a cost when a plasmid arises. It doesn't have, you knock it out, it has no effect on transcription. You overexpress it, has no effect on transcription or no effect on fitness in Pseudomonas. And the domain structure is these domains of unknown function. But if we look through other domains of other proteins which have similar domain structure, there's some hint that they might be involved in phage defense. And so this would be a trade-off, right? Because it would sort of suggest, well, if you have this gene, you're less open to new genes coming in. And that means that you kill phages, but also you've got plasmid fitness costs. And you delete it, and now you become susceptible to phage, but also you find it easier to carry new plasmids. And that could be quite a common trade-off in terms of like openness to horizontal gene transfer. I mean, it's a similar thing with CRISPR, right? That you have CRISPR, you stop phages, but also a lot of CRISPR targets mobile genetic elements, which might be bringing in locally adaptive genes.
Yeah, no, I think that's a lovely thought for chromosomal loci involved in compensation. What about the plasmid loci that are involved, though? Do you think there are trade-offs between which hosts they're in, such that, might there exist hosts where these point mutations now would actually be detrimental for maintenance? Or again, that it's just the naiveness of that plasmid to a host like this?
So again, I don't have any data for this, but I've thought of the experiment that I can do to test it. So what I showed, and I know there's a lot of data that I kind of went through it all like really quickly, and it's really confusing. It's taken me so long just to get my head around all of this stuff. But those PAR genes, so the plasmid mutation causes the upregulation of the PAR genes, which decrease the cost of that particular plasmid, but they increase the cost of another plasmid. Now, if you imagine in a microbiome where plasmids are flying around all over the place, it's quite likely that you end up with both of those plasmids at once. And under those kinds of circumstances, it might be bad to have that mutation. The plasmid might actually increase its negative interactions with the other plasmid and thus exacerbate fitness costs.
All right, so I think that's all the time we have for questions. Once again, thank you, Jamie. Yeah.
Thanks everyone for coming. Hopefully I'll get a chance to visit Moscow, Idaho at some point in the future. I'd really love to come. I've seen photographs, it looks fantastic. And if anybody's coming through to Liverpool, please look me up.